So good afternoon. My name is Sonia Sharma. I'm a tenured associate professor at the La Jolla Institute for Immunology, and I'm going to present my lab's work on the function of ADA2 as an inhibitor of antiviral immunity. So my lab studies innate immunity, and that's the early immune response to pathogens, bacterial, viral, even neoplastic pathogens or tumors. And essentially, what happens is when a susceptible cell is infected with a virus, for example, this is a signal that is recognized by the susceptible cell that initiates a coordinated signaling pathway that results in the secretion of secondary messengers called cytokines by the infected cells. And the classic innate immune cytokine is called type 1 interferon or interferon beta. And this is such an important cytokine because interferon beta has two really important functions in the innate immune system. The first is it acts in a paracrine and autocrine manner to limit the infection in the susceptible cell and the neighboring cells. But IFN also acts in a paracrine manner to alert the immune system and recruit and activate professional immune cells in order to resolve the infection. And these include macrophages. Type 1 interferon polarizes macrophages towards the inflammatory M1 phenotype. It activates dendritic cells, which produce a lot of other inflammatory cytokines, and it even activates B and T lymphocytes. And in this way, we consider IFN beta to kind of set off the entire innate immune response. And so my lab's really interested in gene and protein regulators of IFN beta. And to identify new gene and protein regulators of this pathway, we set up an RNA interference or loss of function genetic screening experiment. And how we do this is we plate stromal cells, in this case, endothelial cells, primary human endothelial cells, in 384 well microplates. We deliver gene-specific RNAi to interfere with the expression of each gene or protein in a well. And after 3 days, we activate the cells with viral DNA in order to activate the interferon beta system. And we can look at different phenotypes of the cells in order to determine if loss of a particular gene or protein increases or decreases the IFN beta pathway response. And we have done this by immunofluorescent imaging, shown here on the bottom, where we're monitoring what's shown in red here. And that's nuclear translocation of an IFN beta pathway protein called IRF3. What we found in our screening results was that loss of ADA2 activated IRF3 and IFN beta signaling. And here you're just looking at the graphical representation of IRF3 activation, which is shown in the dark gray bar. So we showed that in cells in which we knocked down or depleted ADA2 gene and protein, IRF3 activation was increased. Accordingly, IFN beta mRNA and IFN beta protein were also increased in cells which lost ADA2 expression, indicating to us that ADA2 is an inhibitor or a negative regulator of the type 1 IFN beta signaling response that I introduced to you on the first slide. And we obtained similar data not only in primary endothelial cells, but also in other cell types like monocytes and fibroblasts as well. And this is just to show you a bulk RNA-seq transcriptomic analysis to show that without any outside stimulation in ADA2 knockdown cells, there's really a dramatic transcriptomic change. And this transcriptomic change is what I would call a classical type 1 IFN beta response. And you can see that when we look at the types of upregulated genes, which are cytokines, interferon, related genes that are upregulated in ADA2 knockdown cells. So the conclusion here was quite a new one at the time, which was that loss of ADA2 expression activates the IFN beta pathway as well as interferon gene expression programs, which are for the most part considered inflammatory pathways because they activate downstream immune cells. Loss of ADA2 also intrinsically activated the endothelial cells. And here we're looking at the expression of ICAM1, which is a surface molecule that denotes activation of the endothelium, of the vascular endothelium. And this is the flow analysis, and you can see that's quantified here. In cells that were knocked down for ADA2, we saw an increase in ICAM1 expression, denoting an activated phenotype in the endothelium itself. So what's the mechanism? Well, we know that as a metabolic enzyme, the function of ADA2 is to remove or catabolize extracellular purine nucleosides like adenosine or deoxyadenosine, which it converts into inosine or deoxyinosine. And so we reasoned that in the absence of ADA2, that these might accumulate and might activate IFN-beta. So what we did quite simply was here in the dark bars, we treated cells with adenosine or deoxyadenosine or a combination of those with an inhibitor to prevent them from being broken down. And so what we saw was that extracellular purine nucleosides themselves activate IRF3 and also IFN-beta, leading us to the mechanism by which ADA2 inhibits IFN-beta production because it inhibits the accumulation of extracellular purine nucleosides. This can happen in an autocrine manner, but it can also act in a paracrine manner, not only in endothelial cells, but also in monocytes. In this experiment, we just showed that U937 monocytic-like cells react in the same way as endothelial cells do, in that if we treat them with the purine nucleoside deoxyadenosine, they upregulate IFN-beta. We also showed that an ADA2 knockdown endothelium can transmit in a paracrine manner signals to wild-type monocytic cells, in that this knockdown ADA2 deficient endothelium induces an IFN-beta response even in the wild-type monocytes, indicating that these purine nucleosides can act in an autocrine but also a paracrine manner. So what's the mechanism? ADA2 is a metabolic enzyme, so we performed steady-state untargeted metabolomics analysis of ADA2 knockdown cells to kind of tease apart how IFN-beta upregulation was occurring in ADA2 knockdown cells. And what you're looking at here is the untargeted metabolomics data on the right and on the left. And what we noticed immediately was that ADA2 knockdown cells had upregulation of deoxyinosine. And as you can see on the right-hand side, deoxyinosine lies right in the intracellular purine metabolic pathway. And this kind of makes sense that if we're perturbing ADA2, we're perturbing purine nucleoside metabolism. In this case, we observe, as shown in red, an upregulation of deoxyinosine and also its immediate metabolite, hypoxanthine. And this kind of confirmed to us that ADA2 knockdown cells are dysregulated in their metabolism. We also did a very simple functional experiment where we incubated endothelial cells directly with deoxyinosine, and indeed, we saw that this induced IFN-beta mRNA, again confirming that purine nucleosides can activate the type 1 interferon response. We know that from the literature that there's actually a link between changes in intracellular purine metabolism and changes in other metabolic pathways, notably the methionine cycle. And the methionine cycle functions in cells as a metabolic pathway that's very important to produce a molecule called SAM, or S-adenosylmethionine. And SAM is really important because it's required for methylation in the cells, including methylation of genomic DNA. We performed what's called a metabolic flux analysis to analyze the status, the rate of the SAM methionine pathway in ADA2 knockdown cells. And looking at the quantification here, you can see that loss of ADA2 caused an inhibition of the methionine pathway in these cells. What does that mean practically? Well, less SAM means less methylation. Less methylation means that as cells divide, genomic methylation becomes depleted and the DNA genome becomes unmethylated. And that's exactly what's shown here on the right side graph. We looked at in ADA2 knockdown cells, we quantified the degree of DNA methylation after the cells divided, and we saw an impairment of DNA methylation in ADA2 knockdown cells, presumably from the fact that these cells are metabolically dysregulated and have lower SAM methionine cycle flux. This is just another way to show you changes, profound changes in the DNA methylation profile of ADA2 knockdown cells, which we looked at in this case using whole genome sequencing. And what types of genes were demethylated in ADA2 knockdown cells? Well, quite a few, but one of the types that we found to be demethylated were promoters of genes encoding endogenous retroviral elements, or ERVs. And what are ERVs? ERVs are methylation-sensitive viruses that can trigger IFN-β signaling. They're actually old remnants of old retroviruses that are present within our genome. And over the years, as we've co-evolved with these elements in our genome, they have become heavily methylated, which silences their expression. However, it's known that DNA demethylation or hypomethylation, i.e. changes in SAM methionine cycle, can trigger ERV expression. And once these RNAs or DNAs are expressed in cells, they can trigger the innate immune response and stimulate type 1 IFN-β signaling. And indeed, it looks like that's what's going on in ADA2 knockdown cells. What we see in ADA2 knockdown cells is when we examined the expression of dozens of ERV genes, we could see that they were up in the ADA2 knockdown cells, presumably through dysregulation of purine metabolism, dysregulation of SAM metabolism, DNA hypomethylation, and ERV expression. That's a kind of complex metabolic mechanism, but we do feel that we've shown every step of the way that ADA2 knockdown cells have profound dysregulation in their intracellular metabolism. And so this is our model, whereby we think under normal conditions, ADA2 is responsible for removing or catabolizing extracellular purine nucleosides like D8O. And if they're removed from the extracellular environment, they cannot enter cells. And the SAM metabolism, the SAM cycle, and DNA methylation is homeostatic. However, in the absence of ADA2, deoxyadenosine can accumulate. It isn't being removed efficiently, and therefore it is taken up by the cells and metabolized inside cells. And this throws off the normal balance of intracellular purine metabolism, which results in inhibition of the SAM cycle and inhibition of proper homeostatic DNA methylation. When DNA methylation is impaired, ERVs are expressed, and these trigger the type 1 IFN-β expression and inflammatory response. So the conclusion is that we believe that we've discovered in finding a new function for ADA2 in endothelial cells, as well as in immune cells like monocytes, we've discovered a new cellular sensing mechanism for purine nucleosides. Purine nucleosides, it should be noted, are actual inflammatory mediators that are known to be released in damaged cells and tissues and be associated with what's called sterile inflammation. So this isn't a complete surprise, although it was a new observation. And this gives us new understanding as to how these bioactive metabolites can function. I showed you that loss of extracellular ADA2 in primary human cells, and this is a protein which normally removes extracellular D8O, triggers IFN-β expression. The mechanism involved changes in cellular metabolism, and this impacted DNA methylation, causing a hypomethylated state in the DNA that triggers the expression of endogenous retroviral elements. And it's these elements that trigger IFN-β. We hypothesize, or rather we conclude from this, that DATA2 is likely a metabolic disease that affects the endothelium as well as the host immune cells, which are impacted by dysregulated purine metabolism and dysregulated SAM methionine metabolism. This doesn't only happen in human cells. Data from other labs is starting to show that this purine sensing axis may be part of an ancient highly conserved immune system that is present even in worms. For example, data from Emily Tramel's lab at UCSD indicates that purine nucleosides also trigger an ancient conserved anti-pathogenic response in the worm C. elegans. And what Emily's lab has shown is that loss of C. elegans genes like PNP and ADA, which are in the same pathway as ADA2, also trigger what's called a pathogen defense response, similar to what we have observed in mammals. And so I'm going to end there, but I hope that what I leave you with is the idea that ADA2 is an inhibitor of the antiviral response. And the mechanism involves dysregulated metabolism of purine nucleosides and then of SAM methionine. Thank you very much.